Good morning, ladies and gentlemen. We're going to take a trip through the cell today, and first I must tell you about the cell theory. Okay, the cell theory states that all living things are composed of cells. Cells are the basic unit of structure and function, and living things and new cells are produced from existing cells. Okay, as we proceed, we will be entering the cell membrane. The cell membrane is a thin semi-permeable membrane that surrounds the cytoplasm of a cell, which is enclosing its contents. As we keep walking, if you would please follow me, we will be coming up on the centrioles, which are small microtubules arranged in a specific way when two centrioles found next to each other, they are usually at right angles. They are also found and move towards the poles opposite ends of the nucleus when it's time for cell division. Everyone, please stay close and do not get lost. Okay, next is chromatin, which is a complex of nucleic acids and proteins, primarily histons, in the cell nucleus that stains readily with basic dyes and condenses to form chromosomes during the cell division. Cytoplasm is basically the substance that fills a cell containing organelles, vesicles, and other inclusions. The smooth ER acts as a storage organelle. It is important in the creation and storage of steroids. It also stores ions in the solution that the cell may need at a later time. As we proceed with this tour, we come up on the Golgi apparatus. Golgi apparatus is a central organelle mediating protein and lipid transport within the eukaryotic cell. And if we keep walking, we come up on lysosome, which is only in animal cells. Within eukaryotic cells, there's specialized vesicles, which are lysosomes that contain digestive enzymes. These enzymes break down foreign material, which may enter the cell. Now we will be coming up on the mitochondria, which are energy factors of the cells. Mitochondria is different from most other organelles because it has its own circular DNA, similar to the DNA of prokaryotes, and reproduces independently of the cell. If we keep walking, we will come up on the nuclear envelope, which is a highly regulated membrane barrier that separates the nucleus from the cytoplasm in eukaryotic cells. It also contains a large number of different proteins that have been implicated in chromatin organization and gene regulation. Now, one of the most important that we're coming up on is the nucleus. It is the core or the central part around which other parts or groups are gathered. Now we are coming up on ribosomes and free ribosomes. Free ribosomes make proteins that will be used inside of the cell-bound ribosomes are found in the rough yard. Vacuole organelles are fairly simple. There is a membrane that surrounds a mass of fluid, and in that fluid are nutrients or waste products. As this tour comes to an end, active transport requires the input of energy, and there are three types. Endocytosis, which is large amounts of material transported through movements of the cell membrane. The second is phagocytosis, which is when large particles are taken to the cell by endocytosis. The last form of active transport is exocytosis, which is the removal of large amounts of material out of the cell. And as our tour comes to a conclusion, the last thing I will be talking about is passive transport, which requires no energy input from a cell. Okay, I hope everyone had a good time today, and I appreciate you coming to the tour. Thank you.